Good morning, judges. We are from St. Andrews Junior College. I'm Li Wei. She's Eunice. She's Ellery. He's Darius. He's Kit. And he's Lithian. Our project is about using plants' natural ability, phytoremediation, in removing toxic chemicals from the soil. Our aim is to investigate which plants can perform phytoremediation well. This is because we want to use these plants to mitigate the problem of environmental damage through vehicle fumes. As seen from this slide, an estimated 15% of the world uses vehicles. These vehicle fumes contain heavy metals which would dissolve in the soil. In our case study, we are investigating on copper-2, since it is a very common heavy metal found in vehicle fumes. Copper-2 heavy metals are known to be a causal factor of cognitive, mental, and physical disabilities. Phytoremediation is a plant's natural ability to contain and degrade or remove toxic pollutants from the soil and water. As shown in this picture, the plant will absorb the heavy metal and convert it into non-toxic minerals instead. We want to find out which plants can tolerate the heavy metal content best and hence apply this knowledge in our daily life. These plants are capable of performing phytoremediation to be planted near industrial sites or roadsides, helping to purify the environment through natural means. Now I'll hand over the time to Darius Mo.
Study of the uptake of heavy metals in the soil by plants common in Singapore. Different concentration of copper-2 nitrate was added to the different plant samples to test if the plants will absorb copper-2 nitrate from the soil. Positive results were obtained and this shows that it can increase. Now I'll hand over this time to Kit.
Azura is a very popular flowering plant. Mostly they are shrubs and small trees. The flowers appear in clusters and the flower is very small and tubular. It comes in a variety of colors such as scarlet, orange, yellow, and red. The plants are mostly used for ornamental purposes and more importantly because of their medicinal uses. Next, some of India, the plants are grown in tropical regions and are native to Madagascar. It is widely grown as an ornamental plant and house plant, valued for its richly colored, evergreen leaves and thick, irregular stems. While it may reach a height of 4 to 5 meters, rarely 6 meters in natural protected conditions. However, when it is house plant, it is very much smaller. It is slow-growing and upright in habit, tending an oval shape with an open crown. The flowers are small, clustered, and usually white. For bougainvillea, it is a very popular ornamental plant with moss and grown in areas with warm climates. Bougainvillea can be used as a house plant or hanging basket in cooler climates. Tolerance makes bougainvillea ideal for warm climates year-round, and it has a high salt tolerance, which makes it a natural choice for color on coastal regions. Bougainvillea tend to flower all year round in equatorial regions, as where they are seasonal bloomers. I hand over the time to Eunice.
For procedures, we first prepare 4 pots of each house plant. Then we label each pot A, B, C, and D and place them in a shady area. For each concentration, we weigh 0.15 gram of copper-2 nitrate powder using the electronic mass balance. And then after that, we add suitable volume of water to the powder and keep stirring the solution so as to ensure that all the powder has completely dissolved in the water. Each pot of plant will then be watered with 25 cm³ of a specific concentration of copper-2 solution or water every day. Pot A of each species of plant will be watered with 150 ppm of copper-2 nitrate, while pot B of each species will be given 300 ppm of copper-2 nitrate solution. As for pot C, they'll be watered with 450 ppm of copper-2 nitrate solution. As for pot D, it will serve as a negative control experiment in which water will be given to the plant instead of copper-2 nitrate solution. Now I'll hand over the time to Ellery who will continue the procedure.
After 7 days from the start of the experiment, the amount of copper-2 nitrate solution present in the soil is measured. We begin this process by taking 20 grams of soil from each pot. Then, we obtain the soil solution from the soil through filtration. 50 cm³ of the sodium thiosulfate is then added into the soil solution. Measure 25 cm³ of this new solution using the pipette and transfer into a conical flask. Next, add 50 cm³ or 0.01 mole of iodine solution into the beer. Titrate the iodine solution against the solution of sodium thiosulfate and soil solution until the end point is reached.
in which the iodine turns from brown to colored. Obtain the volume of iodine solution used. Then use the volume to calculate the number of moles of iodine solution used. Using calculation, the number of moles of copper 2 ion present in the soil can be calculated. Now I'll hand over the time to Ethan.
On the statistic of isorality, which was suggested in the highest concentration of 450 ppm, adopted 93.2% of the total mole of copper to ion added, while for plant B with 300 ppm, adopted 90%, and B with 150 ppm, adopted 80.8%. By comparing the data collected from count of ethanol and Ogilvy line, it has shown similar trends. Thus, we conclude that as the number of mole of copper to ion added to the soil increases, the number of copper to ion adopted by the plant increases. Therefore, the result shows that all three plants are effectively carrying out phytoremediation. By calculating the percentage of adoption of copper to ion and comparing the plants across the same concentration, we can conclude that Isora is the most effective plant in carrying out the process of phytoremediation. Now I shall invite Li Wei to give a talk on the application of the result.
This method is a better form of energy conservation since it uses natural means to tackle man-made problems, hence lessening the side effects and more cost-effective. These plants will in turn replace the use of man-made chemicals and saving the amount of precious limited resources to produce other goods. Now I'll hand over the time to Darius Mo.
For the conclusion, we can conclude that all of these commonly found plants used are capable of phytoremediation and can remove a substantial amount of heavy metals from the soil without their withering. The results proves our hypothesis correct, as phytoremediation occurs and copper 2 nitrate is absorbed by the plants tested. For the plants tested, Isora has absorbed the highest amount of copper 2 ions among more of the plants. Surprisingly, none of the plants tested had needle, even those exposed to the highest concentration of copper ions, despite the toxicity of copper to plants. For the improvement, we can improve this project by extending the experiment runtime using different heavy metals and different common plants in Singapore. This project can also be extended for removal of heavy metals from both air and water.
I would like to thank our teacher mentors, Mr. Yeo and the lab technicians for their help and their advice during our course of project. And once again, I'd like to thank you all for your attention. Thank you.